So students have informed me there's been some issues with my mic. I'm trying to correct it. So I think I'm going to switch to the big Elvis karaoke mic. And hopefully that will resolve some of the problems. Before, when I would clip the mic down here, people couldn't hear me. When I put it up here, that was causing problems. So hopefully using the big mic will resolve that. Okay, so I'm going to come back to the last slide. When we talked about mitochondria, so what's the whole purpose of this slide is to talk about how do we get proteins from the cytosol into, let's say, a mitochondrial matrix protein, and from the cytosol into the matrix. It's got to go through two membranes. So we talked about how it's initially synthesized in the cytosol, and it's kept in a more unfolded state by cytosolic chaperone proteins HSP70. So that does two things. It prevents it from misfolding in the cytosol. It keeps it in a more linear conformation. And it works with other proteins, which I'm not going to name, to target this protein to the mitochondrial outer membrane. And it'll bind to a TOM protein, which acts as a receptor. It'll move it over, that complex over to another TOM. Numbers are not important. It's just TOMs can serve as receptors. They can also serve as translocons. And so one of the important things is by keeping it in this sort of more linear, string-like manner, it's allowing the protein to transverse through the translocon, very much similar to what we saw with the ER, with, say, secreted proteins or lysosomal proteins. So we keep them in a linear, so that works through a slightly different mechanism with the co-translational translocation. The difference here is that the protein is fully synthesized, but it's kept in a more linear, unfolded manner with the help of these cytosolic chaperone proteins, cytosolic HSP70. Once it gets over to the translocon, the outer translocon, it can transverse through, and we have a translocon of the inner membrane, TIM, which will be in close proximity to TOM because the cristae undulate. And where they come in close proximity to the outer mitochondrial membrane is where we'll see TIM and TOM shake hands, so to speak. Their translocon channels open and allow that linear protein to transverse through into the matrix. And at this point, it's going to have a little bit of help pulling it through with a relative of cytosolic HSP, it's matrix HSP70. They're similar proteins, but they are distinct. This one will have been previously localized to the mitochondrial matrix, and it'll help pull through that mitochondrial protein through the translocons. It requires some energy in the form of ATP hydrolysis. And once it's in there, there'll be a protease which will cleave off the mitochondrial matrix targeting sequence, and now you have the fully formed protein in the mitochondrial matrix. Okay, so that's a very quick recap of what I talked about last lecture. And just to finish off that lecture, we'll talk about another way, another targeting mechanism of targeting proteins from the cytosol into the nucleus. So what kind of proteins could we find in the nucleus? Many types of proteins. It could be transcription factors, it could be histones, it could be epigenetic modifiers, things that have to do with business in the nucleus. Most of those proteins will have a certain sequence called a nuclear localization sequence, or NLS. It can be found in many different parts within that particular protein. So when that protein is translated in the cytosol, it'll be recognized by a protein called important. Not important, important, like import. So what it does is it recognizes that nuclear localization sequence shown in that little black box, and then it'll help guide it. So this is in the cytosol, here's in the nucleus, and here's the nuclear membrane, and there's these large channels called nuclear pore complexes, or NPCs. And the importants are able to bind to a cytosolic protein via the NLS, and then transport, both proteins are guided through the nuclear pore complex into the nucleoplasm. So that's the sort of free-floating goop inside the nucleus. And at that point, now the important, so the important is this little orange thing with a little pointy bit, it has to release that protein. And so there has to be some kind of mechanism to tell it, now let go. In this case, there's a protein called RAN-GTP. And as we get into the signal transduction lectures, we'll talk about G proteins. They're important in many signaling processes. So we will come back to G proteins, and they're named after their ability to bind GTP or GDP. That's one of the reasons they're called G proteins. So RAN GTP, when it's bound to GTP, has a much okay, so yeah, in this case, what happens is the RAN GTP has a much stronger association with the important. So it will now grab onto the important, and that causes a change in the important that it releases the nuclear localization sequence of this imported nuclear protein. So you need, what do we need to get the important to let go? You need to have RAN. It has to be bound to GTP. So if it's linked to GDP, it's not going to bind. So one of the things that helps get rid of GDP and insert GTP is a protein called a GAP, or guanine nucleotide exchange factor. Well, this will come back when we talk about signal transduction. GAPs play a major role in protein signaling. So GAPs are able to help remove the GDP and then insert GTP. So what this is just saying here, it's not hydrolyzing, it's simply exchanging GTP for a GDP or GTP. And so when RAN GTP is in this state, it's going to have a high affinity for the orange important protein, and it's going to cause the important to release the nuclear protein. So now RAN GTP has a high affinity for the important, and now it will be transported back to a nuclear pore complex into the cytoplasm. Now we need to RAN GTP needs to let go of that important, because that important needs to be able to go back into action and find another nuclear protein with an NLS sequence. So in this case, to get rid of GTP, it can be hydrolyzed. And this can be facilitated by what's called a GAP protein, or a GTPase activating protein. It helps facilitate the hydrolysis of GTP to GDP. So now in the cytosol, RAN GDP has very low affinity for important. So it lets go of the important. The important kind of slightly rejiggers, refolds itself a little bit. So now it has a high affinity in the cytosol, much higher affinity for binding nuclear localization sequences found on nuclear proteins. So yes, there's a lot of pieces involved in here, but think about what are the steps that it would take to get a nuclear protein from the cytosol into the nucleus. It's going to need an important. What does the important do? It binds the nuclear localization sequence. That's going to help guide this complex through the nuclear pore complex into the cytoplasm. So that's the first or second kind of steps. Now, once it's in, it's got to have a signal to let go of that nuclear protein. When RAN GTP is present, it has a much higher affinity for the important. Grabs onto the important, shakes it around a little bit, and that makes it let go of the nuclear protein. Now this complex has to go back out into the cytoplasm, but now in order for the important to rebind to another nuclear protein, we need to take RAN GTP and convert it to RAN GDP. And in that case, you're going to need a GAP or a GTPase activating protein to help promote the hydrolysis of GTP to GDP.
So that's your RAN your RAN GDP is kind of useless floating out of the cytoplasm. Does it not need to be imported back into the nucleus?
That's a good question. So if you got lots of RAN GDP out of the cytoplasm, it's kind of a dead duck. Don't we need to get it back in there? The answer is yes. There are a lot of complicated mechanisms that can get it back in. But let's just say yes, you're correct in that we do need to get this back to, we need to be able to get it back into the nucleus, and then we need to get rid of GTP and convert it to GTP. There's a number of complicated mechanisms involved in that, but we're not going to touch it. But good point. Okay, so that finishes up that part of targeting mechanisms to get proteins to the proper place. Now we're going to take a little bit of long. Of course, this is difficult. We launch this in a minute. We're going to talk about proteins that go from the endoplasmic reticulum into the Golgi apparatus. Just bear with me. Okay, there we go. Okay, so far we've talked about proteins that are made in the cytosol, how they get into the ER. And I mentioned a lot of secreted proteins and lysosomal proteins first have to go into the ER. But what are the next steps? Where do they go from the ER? How do they get from the ER out, say, if they're a secreted protein? They're going to have to go through this somewhat unusual structure called the Golgi apparatus. And we'll talk about the steps that it takes to get protein found from the ER into the Golgi. And then from the Golgi can end up going into secretory vesicles. Or it can also go, it's not shown here, into lysosomes. So we're going to talk about those mechanisms today. This is facilitated by transport vesicles, play a really important role. And there's these, what we call sorting signals. And we'll talk about in detail what those are. These are things to help guide a protein from the ER into the Golgi, or from the Golgi to the lysosome, or sometimes from the Golgi back to the ER. We'll talk about some of the modifications that happen in the Golgi. That we already mentioned that, I mentioned last lecture that glycosylation plays an important role, particularly extracellular matrix proteins, help make them sticky. But we'll also talk about how it also acts as a sorting signal itself to direct proteins into the lysosome. And that refers to the mannose-6-phosphate sorting signal, which we'll talk about quite a bit and towards the end of the lecture. Okay, so basically what we're going to focus on quite a bit today is how do we get from here proteins that are found in the lumen of the ER. So we talked quite a bit about the ER and proteins that are synthesized in the cytosol. Co-translational translocation gets them into the ER lumen. And then they're going to move up through these things called vesicles into the Golgi. And then ultimately in the Golgi, they can go one of two destinations, directed towards lysosomes or secreted to the outside of the cell. So before we get any further, what is the Golgi? So it's this unusual structure of disc-like cisternae with no ribosomes. So they're kind of, again, it looks a little bit like the ER in many ways, but there's no ribosomes associated with it. So it's got these kind of almost tubular-like structures, and they have a hollow part in the middle. This is the sort of the lumen of the Golgi. Vesicles will come from different places. In this case, it could be from the ER and confuse with the Golgi. So there's kind of two to get an idea of the orientation of the Golgi. The part that faces towards the ER is called the cis Golgi. The middle part is called the medial Golgi, and then the part that's facing towards the plasma membrane is called the trans Golgi. And so this is a cartoon rendition. This is a transmission electron microscope image showing the structure of an actual Golgi apparatus within a cell. So again, you can see that this part is pointing towards the ER. This part, which tends to be a little more cup-shaped at the top, points towards the plasma membrane. And so proteins that are going to be sorted, i.e. they could be secreted, they could be inserted into the plasma membrane or go to the lysosome. That's where the Golgi plays a really important role. So when we look, I mentioned these things about these vesicles. What are they? Well, when we're referring to vesicles that go from either the ER to the Golgi or from the Golgi to the ER, I'll talk about anterograde and retrograde transport in a minute. But they share they're similar features. So coat proteins play a really important role. For a membrane to kind of pucker up the butt, it doesn't just spontaneously do that. It needs proteins to help facilitate that. There's a couple of proteins that are involved in this. One are coat proteins, as shown in green, and then there's this other protein called the GTP binding protein, sort of this little purple thing. Another important point is what are the types of proteins that we want to get from, for example, if this is the lumen of the ER over to the Golgi. So this is what we refer to as, there's a couple of different types. One could be a soluble cargo protein, or one could be a membrane cargo protein. So these are both the protein types of proteins that we want to get from the ER to the Golgi, and possibly either to the lysosome or to the plasma membrane or secretion. Now, typically what happens with soluble cargo proteins is that they will bind to a membrane cargo receptor protein. And that's also important, as we'll talk about in a minute, whether either it's a membrane cargo protein or a membrane cargo receptor protein, and that binds to a soluble free-floating protein in the lumen of the ER, is that acts as a signal to help recruit coat proteins and also GTP binding proteins. And we'll talk in some detail about those mechanisms in the next slide or so. And then there's one last protein that's important, and it's called snare proteins. So snare proteins actually play an important role in actually facilitating a secretory or a vesicle fusing with another membrane. So if we have we have some kind of vesicle here, and we want it to interact with its target membrane, we're going to have snares. Now, the textbook kind of, sometimes maybe this is a little confusing, but they refer to it as V snares. V snares refer to snares that are associated with the vesicle, and T snares are snares that are associated with a target membrane. It's not really the best designation because these things kind of intermix with one another, and a V snare can become a T snare, and a T snare can become a V snare, and depending on the context of what's happening. And when we're talking about going from the ER to the Golgi or from the Golgi to the ER, I'm not going to get too hung up on, oh, wait a moment, at this point it was a V snare, or oh no, at that point it was a T snare. I think what's more important is to understand what do they do. Snare proteins help facilitate the fusion of membranes. So a vesicle membrane will not just spontaneously fuse with another membrane in the cell. It needs help. And so what happens is these snares twist around each other, and they twist and twist and twist, and they cause the membranes to actually fuse. And there's some energy involved in that as well, which we'll talk about in a minute. So when we're talking, let's for example say this is the endoplasmic reticulum, and that's the cis Golgi membrane. We're trying to get proteins from the ER into the lumen of the Golgi. Therefore, we're going to have the formation of these things called COP2 vesicles. So that refers to vesicles that transport proteins from the endoplasmic reticulum to the Golgi. And we call this form of transport anterograde transport. So from ER to Golgi, anterograde transport. It's a key protein involved in that, those co-proteins, but COP2 co-proteins. We'll talk about the opposite direction, going from the Golgi to the ER. And there's reasons that cells do this, which I'll talk about in a minute. Again, you need co-proteins. In this case, it's COP1 proteins. They form COP1 vesicles, and that's going to help transport proteins from the cis Golgi back to the rough endoplasmic reticulum. That's called retrograde transport. And towards the end of the lecture, we'll talk about how we move vesicles from the trans-Golgi network, the stuff at the very top of the Golgi, to either the plasma membrane or late endosomes. And then there's another type of coat-like protein. It's a little different from coat proteins, but it works together with proteins, called clathrin. Okay, so I'm introducing the players now. Now we'll start to go through the mechanisms involved with how do we get a vesicle, or how do we get a protein from the ER to the Golgi. So we're looking at, for example, COP2 coat proteins. So what will happen is it's either a soluble cargo protein binding to its receptor, or it could actually be, as I'm shown here, a membrane-associated cargo protein. And so there's a little piece that sort of sticks out into the cytosol. And we'll talk in a minute about what kind of signal is found on there. But that little piece that sticks out is going to help promote the recruitment of COP2 co-proteins. And it also requires the co-proteins require a couple of things. One is that you need to get a little bit of a signal from that cargo protein. Secondly, there needs to be the binding of these GTP binding proteins. So that together, the signal they get from the ER cargo protein plus the binding of the GTP binding protein will help cause the recruitment of these coat proteins. And so they start to slowly coat at a portion of the ER membrane and cause it to now bud and form ultimately a vesicle, which pinches, which is released. That's what we call a COP2 coat vesicle or COP2 vesicle. So they're released from the donor membrane, in this case the ER. Now the problem is, if we want to get this vesicle to fuse with another membrane, they can't do it with a coat on. They've got to take the coat off. And so the coat removal will be facilitated by hydrolysis of GTP. It's bound to the GTP binding proteins. Hey, the name kind of makes sense. And so when you get hydrolysis, that provides the energy to dissociate, take off the coat protein, and the GTP binding proteins also are released. And so now this allows the exposure of these little orange lines, those are the snare proteins. So they cannot fuse with another random membrane if coat proteins are blocking their way. Got to get the coat proteins off. So this is, again, I'm going to reiterate some of what I just said, but then also provide a little bit more context, or a little bit more information. So we've got cargo proteins. It could be a soluble cargo protein bound to a membrane receptor, or it could be a membrane-associated cargo protein. It's going to have a small bit that faces into the cytosol. And that's going to act as one of the triggering mechanisms to help promote the association of coat proteins and also the GTP binding proteins. So I'll give another diagram of how this works in more detail in a minute. How do they know that? Because there's a sorting signal. So these cargo membrane proteins use what's called a diacetyl sorting sequence. DXE stands for aspartate, any amino acid glutamate. So those are charged residues. And so they are recognized by the COP2 proteins. That causes the recruitment of the COP2 proteins, and that causes the budding of the membrane. So then it totally forms a COP2 vesicle. We're going to have GTP hydrolysis on the GTP proteins, coats taken off, and now the snare proteins are exposed. And so the snare protein found in the vesicle can interact with the snare protein found on the cis-Golgi membrane. The snare proteins twist around one another, and then that will promote their the fusion of the membrane. Now, to kind of get the snare proteins to recycle, we need to unwind them. This is where ATP hydrolysis is required. So you need the snare proteins to dissociate from one another once you fuse the membranes. That requires energy. That comes in the form of ATP hydrolysis. So we have two energy kind of currencies working here. One, we have GTP hydrolysis. That's referred to those COP2 vesicles with the GTP binding proteins. What's the purpose of GTP hydrolysis in that context? It's to take the coat protein off. That allows the snare proteins to be exposed. They can now interact with snare proteins on their target membrane, the cis-Golgi membrane. They wrap around each other. That helps allow the membranes to fuse together. And then the snares need to kind of like dissociate. The energy currency there is ATP. ATP hydrolysis allows them to unfold, and then they can be, these snare proteins are now integrated into the membrane of the cis-Golgi. And the cargo proteins, either they're inserted into the membrane of the cis-Golgi, or if they're soluble cargo protein, we're not going to get into the mechanisms, they can either remain bound to the cargo receptor or they can dissociate. Now, the other thing is that I mentioned that this doesn't just go one way. You can have a return. The return ticket is called retrograde transport. And that's going to be mediated by COP1 vesicles. So what does that mean? It's basically vesicles that form at the cis-Golgi, and they're going to be helped formed by the recruitment of COP1 coat proteins. It's going to do the same thing that happens here. It's just a slightly different flavor of coat protein, but the same principle is that it forms these COP1 vesicles with coat proteins. There's going to be hydrolysis of GTP, the removal of the coat proteins, and allowing the snares to fuse back down here at the ER membrane. And that's going to return proteins that inadvertently got trapped up in this whole process, which happens a lot, And it's a way to return proteins which are called ER-resident proteins, which I'll talk about in a slide in just a minute. So this is a little bit more like what it kind of looks like, instead of not quite as cartoonish, a little bit cartoonish, but not so much, is when we look at a cargo protein. It's a membrane cargo protein. So it's got a piece that faces into the cytosol, it's got a transmembrane segment, and then it's got a little piece that sticks out into the cytosol. And that's where you're going to find that diacidic sorting signal, the DXE. When it's exposed, it's going to cause a recruitment of the COP2 coprotein, and this also can cause recruitment of the GTP binding protein. So we need both of these players involved to help promote the curvature of the membrane, to promote the bud, and then also have a regulatory mechanism to help remove the coprotein at a later time point. That's where the GTP binding protein comes into play. So let's put this into some kind of context or relevance to, so what? Is this really important? Oh yeah, it's really important because there's various diseases where this doesn't work and be pretty bad. So how many people here have heard of cystic fibrosis? Wow, okay. So it's a very common autosomal recessive disease. It's associated with mutation in a gene that encodes a cystic fibrosis transmembrane conductance regulator, CFTR. And so this is an ion channel which is responsible for the movement of chloride ions, usually from the inside of the cell to the outside of the cell. And so if this ion channel doesn't work, it causes a problem with, let's see, what's the best way? Sort of osmotic effects. So when chloride is exported from the cell, usually you also get the co-transport of sodium ions. And so this can cause, so it's regulation of salt in a way. And if you're not regulating that properly, what can happen is you can get a situation where the outside of the cell is sort of almost dehydrated, and it can lead to a thickening of the outside of the cell, the environment there. This plays a role particularly in the lungs. So what happens is by having this sort of chloride transport deficiency, the cells are not able to osmotically regulate water inside and outside the cell. And then in the lung epithelium, what happens is this leads to the buildup of thick, goopy mucus. It's not watered down, it gets really thick and chunky, and that causes problems. One is it's not as good for allowing oxygen exchange or carbon dioxide exchange. The lung epithelia doesn't work so well, and it's a really nice environment for bacteria to grow. And so patients with this disease, and it does tend to manifest itself in childhood, leads to problems with breathing and then also with bacterial infections. So some crude ways that they still use today to kind of relieve this is they have a big thumper that hangs on the back of these patients and they cough up all this phlegm and stuff. And that sort of helps reduce some of the mucus buildup, but also they have to, quite frequently towards the end, require oxygen. And the only real treatment for this is a lung transplant. So you mentioned there's a mutation. One of the most common mutations is called the delta F508. So phenylalanine at position 508 is missing. That's what the delta means. So it's one amino acid change, and it leads to this disease. Why is that? Well, the main reason is that the delta F508 is enclosed, that particular amino acid is close to where the diacidic sorting sequence is. So it's a transmembrane protein. So it has to get from found at the plasma membrane. That's what it normally does. So under normal circumstances in the ER, it's going to be recognized by COP2 coproteins. You're going to have those COP2 vesicles. They're going to move from the ER into the Golgi, and then they'll be processed through the Golgi and ultimately go through the secretory pathway, through secretory vesicles, will go up to the plasma membrane and integrate into the plasma membrane, and then allow the proper conductance of chloride ions. Now, the delta F508, what's odd about this is that the chloride ion channel is still completely functional. The problem is that it can't get from here to there because the delta F508 mutation causes a change in the structure of the protein such that the diacidic sorting sequence doesn't stick out into the cytosol. As a result, those COP2 proteins can't bind to it. And as a result of that, you get the mutant form of the protein, it just gets stuck in the ER membrane. It's all dressed up, it's got no place to go because it doesn't have the ticket out there to help get the COP2 proteins to kind of latch on and form the vesicle to help move it into the Golgi. So the important take-home message here is a protein that's a cargo protein that has to get from the ER into the plasma membrane. It has to have a functional diacetyl sorting sequence. The sequence is there, it's preserved, but the delta F508 mutation causes the structure of the protein to change such that that sequence can't be exposed. It can't face into the cytosol, and it can't interact with the COP2 proteins. You can't get those vesicles formed, and therefore there's no CFTR going up to the plasma membrane. Some of the strategies that we're trying to employ is some sort of trick to get the diacetyl sorting sequence to be exposed. Because if you get the delta 508 mutant form up to the plasma membrane, it can function as a chloride channel. It just can't get there. All right, so we've talked quite a bit about anterior grade transport. Now we're going to talk about retrograde transport in COP1 vesicles. Where does this play an important role? It plays an important role in what we call retrieval of ER resident proteins. What are ER resident proteins? The same proteins I talked about last lecture. So they are proteins that have a function in the ER. Chaperone proteins, BIP, or calnexin, or calreticulin, or protein disulfide isomerase. So all those proteins that help with the proper holding proteins in the ER, sometimes when these COP2 vesicles are made, they inadvertently get kind of caught up. They're an inadvertent stowaway, and they end up getting transported up into the Golgi. So once they're in there, they really don't have a functional purpose. That's not where they normally function. They need to be back down here. This is where the environment they normally work in. It's also where the environment in terms of pH is where they work most effectively. So how do we get these ER resident proteins back down to the ER? Turns out that they have a sorting signal as well called the K-del sequence. It just refers to lysine, aspartate, glucine, and leucine. So K-del, it's found on these ER resident proteins like VIC, BIM, calnexin, calreticulin. And it will bind to a receptor found at the Golgi membrane called the K-del receptor. And it has a high affinity for this K-del receptor, so that little kind of like seat-looking thing. It has a higher affinity here because there's pH differences between here and here. And one thing you'll see is when we go from the ER to the Golgi and then on to other structures like the endosome, is that the pH drops. What does that mean? There's more hydrogen ions or protons in the Golgi. So the pH is a little bit lower in the Golgi when you compare to the pH found within the lumen of the endoplasmic reticulum. And so at this lower pH, the K-del peptide, or the sequence on this ER resident protein, has a high affinity for the receptor. It's going to bind to that receptor, cause a little bit of change in the receptor, such that it kind of now pokes out a sorting signal. And that sorting signal is, we sometimes refer as a dibasic. So KKXX sequence. It's not shown here. It's not really the best diagram to show this because it implies that it's just this flat protein. No, there's a little piece that sticks out. It's not shown in the picture. So that little piece that sticks out is going to contain that dibasic sorting signal. And so that sorting signal will cause the recruitment of COP1 proteins. Same kind of principle. It helps bud the membrane, and then it'll pinch off as a COP1 vesicle. And again, same kind of steps as we talked about before, hydrolysis of GTP, removal of the coat proteins. The snares can interact, it's not shown here, and that'll cause the fusion of that vesicle to the ER membrane. It releases the protein So the KDEL receptor, when bound to the KDEL containing protein, has a low affinity in the ER because it has a higher pH. So it's a pH, and we'll see this repeat itself a few times in the next lecture or so, is that pH can dictate how proteins bind to one another. Some pHs are permissive, some pHs are not. So low pH is a permissive pH that allows the KDEL peptide to bind to its KDEL receptor. That's going to help trigger activation of the receptor to expose its dibasic sorting signal to cause the recruitment of the COP1 proteins. That helps form that vesicle to help move that particular protein back down and fuse. The vesicle fuses with the ER membrane. And the pH is a little different, it's a little bit higher. There's not as many protons or hydrogen ions there. And under those circumstances, the KDEL receptor and the KDEL containing protein, they don't bind, they dissociate, and now we've returned that ER resident protein back to where it belongs, in the lumen of the endoplasmic reticulum. All right, so those are kind of the main things to focus on. So A, why do we need this? Because ER resident proteins. What ER resident proteins? BIF, calnexin, calretictin, proteins of sulfide isomerase inadvertently get transported into the Golgi. We need to get them back. Those proteins will have a KDEL sequence, and then we just talked about pH dynamics and a KDEL receptor. Another sorting signal, KKXX, the dibasic sorting signal, is going to help cause recruitment of those coproteins and formation of that vesicle, and which ultimately will fuse and return that protein back into the lumen of the ER. It releases the KDEL receptor and the KDEL containing protein because of the higher pH. That's basic. I've said it a couple of times, hopefully that resonates. Now, we talked about protein glycosylation. It happens at many stages. It starts in the lumen of the endoplasmic reticulum. We talked about Vesicular transferase last lecture. There's enzymes that are similar that are found in the Golgi, many of them, and they're referred to as glycosyl transferases. So they add sugars. And then there's other enzymes that help remove it. Those are called glycosidases. So I'm not going to get you to remember, well, you know, you have an abundance of linosidases here, and you've got some transferases, and blah, blah, blah. Who cares? It's just important to know that modification of carbohydrates stuck onto proteins, either adding more or taking off, happens in the Golgi. And there's a sort of enrichment at different parts of the Golgi for certain types of glycosidases or certain types of glycosyl transferases. I'm not going to ask you to memorize all these names and where they're found at different stages, because it doesn't really matter. But it will come I will ask you to focus on a few as it relates to how do we get proteins, maybe at the top of the Golgi, so the trans Golgi, that might end up going to be secreted, or more specifically, end up in the lysosome. The lysosome is a really important organelle. It's filled with things called acidic hydrolysis, and they help break down all sorts of problematic material in our cells. It could be lipids, it could be proteins, and they function at a low pH. So they need to get into this structure called the lysosome. And this is those key enzymes are going to be moved from the trans Golgi to the lysosome through an intermediate organelle called the endosome. And this is going to require other types of coproteins. One is called adapter protein, or AP. So adapter protein coat proteins will promote a little budding at the trans Golgi membrane. And it typically needs the help of another kind of unusual protein called clathrin. So clathrin forms in this sort of three-legged structure called a triskelion. And then the triskelions all interact in a unique manner. And they end up forming these sort of almost hexagonal structures. So when you think about a soccer ball, that triskelion, when they interact in such a way, form these structures which cause basically a ball-like structure. So it's an unusual property of clathrin. They associate as a three-membered structure called a triskelion, and then those triskelions associate with one another. So you can see that red one, there's a yellow one, and there's a green one. So by forming those complex structures, they form almost like hexagonal structures that self-assemble like a soccer ball. So, getting back to how we get a lysosomal, a protein that's destined for the lysosome. Where did it start? Well, it started initially being synthesized in the cytosol, just a little bit, and then it was recognized by a signal receptor, because it has a signal protein, a signal sequence. It's going to go through co-translational translocation into the ER, and then that is ultimately move my anterior grade transfer from the rough ER to the Golgi. It's going to move its way up in the Golgi and then finally get up to the trans-Golgi, and there it needs to know how to get from there to the lysosome. And so there has to be some kind of sorting signal. In this case, it's a sugar called mannose-6-phosphate. And so this mannose-6-phosphate, when it's put onto that lysosomal enzyme, is going to be recognized by something called a mannose-6-phosphate receptor. And that will help cause recruitment of adapter protein and clathrin to help form a vesicle to transport it from the Golgi, trans-Golgi, to ultimately, there's an intermediate, but ultimately the lysosome. So before we get into that though, how do you put that mannose-6-phosphate on there? So remember, that protein, that lysosomal enzyme, when it went into the ER, when it's co-translationally translocated, oligosaccharidotransferase put that big branch sugar thing on it. And maybe it went through the Golgi, got a little bit shaved off, a little bit added. But the added part in particular that's really important is that we need to put on a phosphate group onto one of the mannoses. So those little blue dots are mannose carbohydrates that's been previously stuck onto the lysosomal enzyme. And in the trans-Golgi, in the cis-Golgi, I was a little off there, it's going to be recognized by this enzyme called N-acetylglucosamine phosphotransferase. It's a big mouthful. Sometimes they call this glucmac. That's a little easier to say. So what it does is this transferase has a recognition site for the lysosomal enzyme. So there's certain amino acids found on the lysosomal enzyme, shown in those little red circles, which will be recognized by this enzyme. It also will bind to a pre-existing phosphate sugar structure. So it has glucosamine, N-acetylglucosamine with some phosphate stuck on it, so it binds that. And so what this does is it links the lysosomal enzyme with those pre-existing mannose sugars stuck on it with a phosphate group. And so it'll put this phosphate group onto a mannose, and there's a little bit left over, and that's the N-acetylglucosamine. It's another carbohydrate, and a phosphodiesterase will cleave that little sugar off. So you end up with a phosphate stuck on mannose. That's the mannose-6-phosphate. So this acts as a signal. So just to reiterate what's happening, we got a protein that we ultimately want to get to the lysosome. It's had to go through the ER. It had to have that kind of complex sugar thing that's stuck on it in the ER. It gets transported into the Golgi. And there it's going to be recognized by the N-acetylglucosamine phosphotransferase. How will it recognize? Because there's a recognition site on that enzyme that will recognize certain amino acids found on that lysosomal enzyme. All lysosomal enzymes have those certain amino acids, which will be recognized by this phosphotransferase. The phosphotransferase also will bind to this phosphate structure, which also contains N-acetylglucosamine. So it's a sugar, a square sugar, stuck to some phosphates, and some other stuff I don't care about. So it sticks on that phosphate and that little bit of sugar onto a mannose, and we need to get that little extra sugar off, the N-acetylglucosamine off, and that will be achieved through the phosphodiesterase. Now we have the mature lysosomal protein. It's got the correct signal, the mannose-6-phosphate stuck on it. So in this case, as it moves up, so the mannose-6-phosphate receptor is down on the lysosomal enzyme and kind of moves up to the trans-Golgi section, and now it's going to be recognized by the mannose-6-phosphate receptor. It's that little sickle-looking thing. So it's going to, it binds to it, it recognizes not the protein, but that mannose-6-phosphate. The carbohydrate with phosphate stuck on it. So the mannose-6-phosphate will bind to it, causes the change, which causes recruitment of adaptor proteins and the clathrin triskeleton. So they now form this clathrin-coated vesicle, and there's removal of that through some mechanisms we don't need to get into, and it will form this uncoated transport vesicle, which will fuse with something called a late endosome. So we're going to focus just on this. We're not going to talk about this part. It's another thing. We will cover an aspect like it when we talk about receptor-mediated endocytosis. So we're only going to focus for this lecture on this part. So we've got to the stage where this transport vesicle will fuse with the late endosome. Now, at this point, the mannose-6-phosphate receptor is going to dissociate from that lysosomal enzyme. Why? Again, a pH thing. So when we're here, it's slightly acidic, so pH 6.5 in the Golgi network. But the late endosome actually is more acidic. There's mechanisms to pump in protons to make it more acidic. And so the pH drops down to pH 5.5, and under those lower pH conditions, now the receptor cannot bind to that lysosomal enzyme or bind to the mannose-6-phosphate residue found on that enzyme. So it dissociates, and then the late endosome can fuse with a lysosome, and now that enzyme will be functional within the very low pH environment of the lysosome. And so the mannose-6-phosphate receptor can also be returned back into the to the Golgi, or sometimes it can go up to the plasma membrane. Not so important. I think I'd rather focus more on the mechanism of getting a lysosomal enzyme to the lysosome. So you got to take a step back and you can think about, hey, it's got to start in the cytosol, and it's got to get into the ER. It's got to be modified in the ER. It's got to make it to the Golgi. Then it has to be modified further, the N-acetylglucosamine phosphotransferase, which will then put on that phosphate group on the mannose. And then at this particular pH, it's going to be recognized by the mannose-6-phosphate receptor. At that pH around 6.5 binds really well. It's going to help cause the recruitment of co-proteins, adapter, AP proteins, and clathrin. That's a little different from when we talk about the Golgi-ER connection. We're not talking COP1 or COP2, we're talking adapter proteins. And they work with another unique protein, the clathrin triskeleton. So that will form that clathrin-coated vesicle. It needs to be uncoated through an energy-dependent effect, which I'm not going to test you on, it's not important. And that allows fusion with the late endosome. Again, pH dynamics predict or will dictate how this interaction occurs here, but does not occur here because of the lower pH. And then finally, why is this important? Because if you can't get the right kind of lysosomal enzymes to lysosomes, all sorts of problematic material that can build up, particularly when your brain doesn't get degraded. And so these are called lysosomal storage diseases. You're not able to break apart problematic material in lysosomes. And one example of this is called inclusion cell or I-cell disease. It's one of the most severe forms of lysosomal storage diseases. In this case, they don't have a functional N-acetylglucosamine phosphotransferase, can't put that signal on, and then the lysosomal enzymes don't end up going to the lysosome, they end up being secreted. And, I got 1 minute, let's finish this. So, glycolipids and other problematic material build up in the lysosomes, and those glycolipids, particularly in the brain, if they don't accumulate, they can steal potentially toxic molecules like reactive oxygen species, and it causes destruction of neurons. So this one is quite severe in that they have severe neurological abnormalities at birth, and Germany don't even make it into early childhood. And there we go, I did it on time. Just make sure I shut this up.
I have a question about cake sorting simulates and from my understanding, this was like expectations transport and the code transfer. Is that because the code transfer you don't have to instead you use energy because for the cake sorting signals, don't use energy.
Well, there would be energy in the form of GTP hydrolysis to remove one. So I'm just going to get out of the way for the next instruction.
But it's also isolated
Well, energy in the form of GTP hydrolysis, right? So there's a couple of different
I'm Reed.
Hi, Steve.
I'm a citizen bill.
Okay.
I'm running for ACE as the state president, and I'm going around classes and giving like a 1-minute presentation about that.
Unfortunately, no. I get a lot of requests, and so if I gave time to everyone, I would have enough time